Marsden. She is a professor in the Rubenstein School of Environment and Natural Resources at the University of Vermont. Ellen received her master's degree and PhD from Cornell University, and then served as the director of the Lake Michigan Biological Station, Illinois Natural History Survey for 6 years. She has been on the faculty of the University of Vermont since 1996. She has conducted research on bird migration, evolutionary genetics of frogs, genetics and early life history of lake trout, alternative control methods for sea lamprey, ecology of exotic species, population dynamics of lake whitefish, and constructing artificial reefs to enhance lake trout spawning. Ellen has worked in each of the Great Lakes, but her work is currently focused in Lake Champlain, where she has established an acoustic telemetry array to track fish movements, where she's evaluating the effects of lacustrine habitat fragmentation on fishes, and is documenting the recovery of naturally reproducing lake trout. Thank you, Ellen, for sharing your knowledge and experience with us today. Please go ahead and take it away.
Great. And I trust you can see my screen?
Yes.
Perfect. Well, thank you very much for this invitation. It's always fun to, particularly to give talks with a Sea Grant audience, because I know people are interested in the science and also interested in the application, which is part of what I want to stress today. So this is my title, Fisheries Research. And fisheries research, of course, is all about the people who you're doing it with. No research is done alone. And so this is the team. I'm just the person who gets to talk about it, but these are the folks that really do the work. The undergraduate students, graduate students, postdocs, and of course, the captain and crew of our research vessel, who, by the way, is in our last year. We will have a new research vessel, and that's something completely different we could talk about. A new, very, very high technology vessel will be launched next year. So it's all about the teamwork and the collaborations. And I really want to emphasize that, not just the people who are doing the work, but the incredible relationship we have, particularly in Vermont, a nice small state, where the state agencies, the federal agencies, and the university very much work together, the researchers communicating with the managers and affecting management and filling management needs. So briefly, let's go back to Lake Champlain. Presumably a lot of you, maybe not all of you, are very familiar with this lake. It's small. Well, if you grew up here, you might think of it as large. If you grew up with the Great Lakes, it's really very small. It barely shows up on the map, but it has attributes that make it of particular interest. It's small enough that we can think of it as a mesocosm. You could do research throughout Lake Champlain within a week. You could hit the whole lake. Whereas working on Lake Superior, for example, it might take you 24 hours just to get to some of the sites, let alone getting back. That would take another 24 hours. So we think of this as a Great Lakes mesocosm. Mesocosm meaning a small experimental version of the big thing. And so we can answer a lot of questions that are relevant not just to Lake Champlain, but to the Great Lakes as a whole. It's also fragmented in that the figure to the right there just shows that fragmentation. The different basins have been historically isolated by the construction of causeways starting in the 1850s, such that we have discrete basins that each have rather different types of productivity, slightly different fish populations, and slightly different issues related to them. So it's like 4 lakes in one, and that makes it, again, particularly interesting to do research. The lake had a commercial fishery, as the Great Lakes did, but it closed in 1917. It was deemed that the lake would serve anglers and the tourist industry better than it was serving the few farmers who did a bit of commercial fishing on the side. The primary fisheries then and now are still Atlantic salmon, walleye, lake trout, basses, and perch. And here I'll be really focusing just on the cold water fishery, Atlantic salmon and lake trout. But of course, Atlantic salmon and lake trout that are in the lake right now are all, or most of them now, originating from hatcheries. They've been stocked because both of them were extirpated from the lake by 1900. The Atlantic salmon disappeared due to the building of dams, which blocked their access to places they wanted to spawn in rivers. But lake trout, we really don't know why they disappeared by 1900. So management comes in, and management says, wow, lake trout and Atlantic salmon filled very important needs, both for the fishery and for the lake. And so they made the decision in the 1950s to restore salmonid populations, with the focus at that point in time was to create a fishery. They really weren't thinking about ecosystems. The word had really basically barely been developed. They were thinking just about the need of people who wanted to have fishing opportunities. So to restore the fishable populations, they began stocking. And at that time, they were putting an average of about 160,000 lake trout and about 700,000 Atlantic salmon, starting on a regular basis in 1973. Why so many Atlantic salmon? Because they live a much shorter lifetime. They live to 7, 10, 12 years old, whereas a lake trout can easily live to 50 plus years. And also the Atlantic salmon were also stocked at 2 life stages, where survival would be much lower than the life stage at which lake trout were being stocked. As they were stocking them, we said, wow, we need to know a little bit more about the lake than just how many predators that are there out there. Do we know whether the lake will support this many predators being stocked? Are there enough forage fish, the food that those predators will eat? So the state, that is Vermont, started that survey in 1982. And then they started assessing the progress of restoring the lake trout and Atlantic salmon populations by going out to spawning sites. There were 2 spawning sites, one at Whalen Bay in the south, one at Gordon Landing in the north, to look at how these fish were doing, how many fish were showing up at the spawning sites, were they growing well, were they a good size at their age, did they have good weight, did they have good condition, and was there any evidence that there were wild fish? Were any of the fish unmarked? Have to remind you that every fish that's put in the lake by a hatchery has a mark. In our case, it's a fin clip, and it's done on a 5-year rotation, depending on which fin they clip, so you know which age fish that you just caught. If it had a right pectoral fin missing, which year was that fish likely stocked? The problem was, of course, that as they were assessing those fish during the spawning period, they were finding continuing evidence of really severe parasitism by sea lamprey. And of course, you only see the fish that got wounded but lived through it. This is a particularly small fish that has a lamprey wound, but the fish was still alive when it was caught. What you don't see is all the fish that, as a result of lamprey wounding, died. And that, of course, is affecting the success of the stocking program, particularly when you compare the wounding rates in Lake Champlain compared to the Great Lakes. They were 5 to 20 times higher than what was seen in the Great Lakes. It was really, the Great Lakes people would say, it's amazing you even had any live lake trout or Atlantic salmon in the lake. So the decision was made logically to start doing something about seeing if it was possible to control or suppress sea lamprey populations. And that began in 1990. And with that, of course, came the concern that if you can suppress the sea lamprey populations and the wounding declines the way they wanted it to, and the way, in fact, they successfully did, and when I say there, this is the state and the US Fish and Wildlife Service, again, working cooperatively to do the work. If you increase the survival, then you're going to increase the pressure on the forage fish populations. So they said, well, let's be cautious about this. And they decreased stocking of lake trout to 84,000 per year and slightly decreased the amount of salmon going in each year. But these assessments that were going on every year, those are the 2 sites at Gordon Landing and Whalen Bay, began to show a lower wounding rate. There was indeed a successful suppression of sea lamprey, but no evidence that those wild fish that had been stocked as little fingerlings, little age 0, age 1-year-old, that were surviving well and showing up at spawning areas, there was no evidence that they were producing any naturally produced fry. So that's an interesting question for managers. So this is about the time that I joined the team in Lake Champlain in 1996. And because I'd been doing research on lake trout in the Great Lakes, began doing it here. And the first logical question is, wait, all these fish that you see out there came from hatcheries. Do they know where they should be spawning, are there enough of them showing up at spawning sites? Remembering again that only 2 of these sites had actually been identified at that point, and in fact there were adult fish showing up, the stocked fish. So we know there's all those fish showing up. They're showing up in huge numbers at these 2 spawning sites. So there's no paucity of adult lake trout that know they should spawn at these kinds of sites. And then we said, well, are they spawning successfully? Are they doing the right thing, so to speak? Yes. Egg densities were very high, and lake trout spawn in November, so it's a kind of a cold time of work to be year to be doing this work, but lots of evidence of successful spawning. They don't hatch until the following year in April to May, and so 6 months later we go back out. It's still pretty cold. It's this time of year, and we found that in fact there were lots of little lake trout fry being produced. So great, there's no problem here. We've got good spawning sites that the lake trout find. Those spawning sites can support successful egg incubation. The water quality and the habitat is good. We've got lots of little fish like this, but no evidence that they get through their next winter of life. So they hadn't been showing up at the spawning sites. And when we at the university started sampling for little fish beyond the first year, what we'll call recruited fish, age 1 and 2 and 3, we didn't find any. We found juvenile lake trout, but all that we found were stocked fish that had fin clips. So there's a bottleneck. Something's going wrong, that these fish can produce little fry, but those fry do not survive. So in 2015, a number of years later, with a new captain who had done this kind of work on Lake Ontario, I'd worked with him back then briefly, we said, okay, Steve, can we start up some sampling and sample juveniles again, more intensively, more focused with techniques he was familiar with, and see if we can in fact find more juveniles and get a better sample size to assess potential natural reproduction. And the first trawl we put down with this, as I say, captain with a great deal of skill, we found lots of juvenile lake trout. Like, wow, that was impressive. A really high catch, but even more exciting was, of those fish, we had a lot of them that were in fact unclipped and wild. Now, this was in fact new. As I say, we had found juvenile lake trout before. We hadn't found any unclipped ones. And if this had been happening all along, this production, this survival of these wild fish, we would have eventually seen it in the fall when they were 6 years old and getting ready to spawn. That's how long it takes them to mature. We would have seen it, and we hadn't seen it. So this is something new. So what we got was that first year, we had about lake-wide, over all our sampling sites in 2015, about 23% of them were wild. And I won't go into all of the details. There was a lot of stuff going on here. We had very different distributions of abundance of juvenile lake trout in the south portion of the lake, the middle of the lake, and the north. And we also had different ratios of wild and stocked fish. Here, I'm just going to show you the proportions of wild fish in the central lake, where we had the most juveniles in general. So that first year, we had almost 30% of them were wild. So, sort of from 0 to 30, wow, abruptly showing up in this juvenile population. So we kept doing it. Now, the numbers on top of each of these bars are the number of fish that we sampled. So you'll see those numbers going up. That's not because there were more of them, but we were putting more effort out there. To really understand abundance, you have to know the abundance per unit effort. We went out a lot, we caught a lot more fish. But the next year, in terms of the height of the bar, we've got 45%. So the number was increasing, meaning that there were more year classes, more years worth of fish entering the population. The trawls that we're using only sample 4 age classes, age 0, 1, 2, and 3, and after that the fish are too big and they escape the trawl and we don't catch them. So we're watching new ages move into the population and then move back out of the ability to see them in the trawl. Wow, 2017, this just got amazing. Now we've got about 75. It keeps going up, just a little bit in 2018. About 76% of the juvenile population is wild fish. And we know it's an increase in wild fish because they still keep stocking the same number of stocked fish. So the difference is this addition of these wild fishes. 2019, the numbers went down a little bit. wow, not enough to worry about. There's going to be variation. The fact that it kept increasing for 4 years in a row was astounding. This is more normal. Things will fluctuate. And then 2020, well, it's a slightly bizarre year, as we all know. COVID really put a dent in our effort. We didn't even get the boat operational and were allowed to use it until July, which is halfway through our sampling season. And then we were also training new crews, and everything was different. So that number has to be taken with a great deal of a grain of salt, so to speak. But we've now got a population of fish that is being dominated by fish being produced in the wild, not those that are being stocked. This is success. In the Great Lakes, there'd have been, cheering all over the place. I mean, we were cheering. If you heard us doing happy dances out on the Melisira, that's what was going on. So success. What do we want? We sort of, hand in our trawl and go home and rest on our laurels? Well, no, not yet, because these are just the juvenile fishes, and they weren't yet showing up as spawners. They've got to survive to age 6 before they actually become part of the reproducing population, the mature population. So we have to keep following the population. They're not there in the fall, they are there as juveniles. So we started gill netting in the summer as a way of catching those larger fishes. And the state was doing the same thing. Again, this collaborative, we're constantly sharing results with the state, and they're sharing their results with us. They're saying, we see nothing in the fall, let's go out in the summer and we'll all sample the summer fish and see if they're showing up in the summer. And whoop, yes, absolutely. By last year, we were seeing a quarter of the fish that we caught as adults and larger juveniles being wild. So they're clearly surviving. Why aren't they showing up at the 2 spawning sites that are being sampled in the fall? Well, there's 2 possibilities. One, of course, is they're still growing up, so not enough of them have quite grown up enough to be mature and start showing up at spawning sites. But we've got a lot of fish out there, so there's something else going on, and that might be partly the particular sites that we're looking at to determine where these fish are reproducing. Maybe we're not looking at the right sites. But before I get to that bit, I want to bring up something else, and that is we've now got at least 9 year class. A year class is the group of fish that were spawned in a particular year, right? So there's 9 ages of fish out there. That's a lot of fish, and they're getting bigger, which means their appetites are growing. We start to have to worry again about the forage abundance. Now, unfortunately, for a number of reasons, the state, that is Vermont, stopped the forage fish survey that had been going on for about 33 years. They stopped it in 2015. Feeling that it wasn't giving the right kind of information or sufficient information, and it was a lot of work. So just at the time we start seeing this extra pressure on the forage base, we stop having information about the status of the forage base. Now, what do I mean by forage base? Largely, it used to be these smelt. Rainbow smelt are a native species in Lake Champlain and were the basis of most of the forage. And then also these little slimy sculpins, which are a bottom fish that are not normally seen much by anglers. Alewife invaded in 2003, so they got added to the potential consumable fish. And we've also got this little fish, also very obscure, not well known. It's out in deep water. It's about 1/3 of our catch when we're catching fish in a trawl, because we catch all of these fishes while we're catching lake trout. And these fish are just an interesting mystery that we're still scratching our heads about, because we don't see any fish eating them. So somebody out there is eating them, but we don't know who yet. Well, what do I say? When I say predators, it's not just about lake trout, because we've got the Atlantic salmon, also dependent on these 4 species in different ratios. We've got walleye, major predator, eating these fishes, particularly smelt, alewife. And then we've got burbot, the freshwater cod, which is not commercially fished in this lake, but is in the Great Lakes, and is also a major predator in cold, deep waters. What drives the forage base abundance? Well, everything, the productivity of the lake, which is affected by phosphorus inputs that drive the productivity of plankton. And the productivity then gets translated into foodability through the food web of zooplankton, and then small fishes, and then larger fishes, and then predaceous fishes. We've got spawning habitat that's also affected by watershed inputs in terms of sediments affecting habitat quality. course, we've got two kinds of predators, those that are there anyway, the walleye and the burbot, and then those that are being stocked, and now the wild lake trout. We've got water temperature changes during global climate change, and we've got invasive species. It's a really complicated system to really understand what's affecting how much forage is out there. So to really understand what's going on out there, what we need is a model that brings in all of these components. And we're in the middle of trying to create and produce a whole lake food web model that brings in all of these pieces of the food web, the actual biota, and then also starts to bring in things like watershed inputs and temperature effects and chemical effects like phosphorus. It's a heavy lift. We're about 2 years into it, still not quite there, but making significant progress. Ultimately, what we'd like to be able to do is say, we only have control really over two portions of the food web, two portions of the food web. One is this: we keep stocking predators. That's an input that is not responsive to what's happening externally until you have a lot of information. So we just keep on stocking 84,000 lake trout and a whole bunch of Atlantic salmon. And then we have inadvertent input on the food web by watershed inputs that many of you will be very familiar with, things like sediments and phosphorus coming off farmed fields that are then affecting the productivity and algal blooms. So ultimately, a food web model would help us predict what would happen if we dropped phosphorus, what would happen if we increased or decreased stocking. Can we actually understand responses of the forage base and then of the predators? So to do that, one of the pieces of information we need is a modern forage fish survey. We need new current information about the forage base. And part of that means redesigning the forage fish survey. The one we had been started back in '82. In the 40 years since then, we've learned a huge amount about how to do forage fish surveys. And largely, again, this is collaboration, but now with our Great Lakes colleagues, particularly the US Geological Survey, Fish and Wildlife, state agencies. They've been doing this for the last 40 years, and in the process, learning a great deal about when to sample, how to sample, where to sample, how often to sample. So we're rewriting, if you will, the forage fish survey for Lake Champlain with the idea that ultimately what we want to know is, what is this predator-prey ratio? Are there enough prey for the number of predators? The predators are being assessed one way, the prey are being assessed another way with the forage fish survey. The forage fish survey should tell us what's the composition. For example, are alewife displacing or competing with smelt? Are we losing smelt in favor of alewife? What's happening to our slimy skelpin? What's the relative abundance? Are we staying stable in terms of the number of each species? Not just the number, but the size of them. Are they growing well? Do they have good condition, i.e. weight, such that we have the ultimate metric we really want? We don't want to know number. Ultimately, we want to know biomass, the total weight of forage fish that's out there that's going to be feeding the total weight of predators that are being stocked and are also naturally produced in the lake. So that starts this year. We did some preliminary work last year, and we'll be out as soon as the boat is in the water doing this sort of work, counting a lot of fish. Meanwhile, back at the lake trout, we've got all these lake trout that are being naturally produced, that are growing up, but we still don't know why. For 30 years of my career, I've been asking, why aren't they surviving and reproducing? Now the question flips on its head and going, why are they surviving and reproducing so successfully? Well, one of the hypotheses I mentioned comes back to where we've been looking and where all the reproduction that we know of has been happening. So each of these stars indicates an area, not just that the state has been surveying, this is Gordon Landing and Whalen Bay, but sites that in work with graduate students starting in 1999, we looked to see are there other places where they spawn. And basically every place that we looked by diving in with robots and with sampling that looked like they might be good for lake trout reproduction in terms of good habitat, the lake trout knew that, and the lake trout was spawning there. Great, lots of sites of spawning. But look at them, they're all near shore. And we've added a few more stars on the New York side since this study. They're near shore. Well, is it that lake trout are spawning near shore or that we're looking near shore. Keep in mind, lake trout are spawning in November. That's when the gales of November come in. I don't really want to be working out here in the middle of a November storm in deep water, in cold water, trying to find a proverbial needle in a haystack on offshore reefs. And so we've never looked out there. Meanwhile, in the last 20 years, a lot has happened in the nearshore areas. They're subject to a lot of erosion during storms and flood events, and erosion basically is very bad for spawning sites. And they've also been colonized by zebra mussels that came in 1993. And so the areas we look are completely coated in dense colonies of zebra mussels. And they're not very good for spawning habitat quality because they block the ability of the eggs to penetrate down into spaces where they'll be protected over the winter. So we started to ask the question, what about what's happening out there? Out there, offshore and in deep water, are sites where the zebra mussels may not be as dense because they're not as, they like shallow water, not deep water. So they're more likely to have, they're less likely to be colonizing reefs in deep cold water. And the sediments aren't as bad out there. The sediments tend to settle near shore first. So what if we've been either missing offshore spawning all along, or it has started to occur more than it used to, simply because these lake trout that were spawning inshore are going, these sites are kind of messy, let's go find someplace else. So it could be either we've missed it or they've started potentially spawning out there. Are these sites successful places for lake trout to spawn, given the stuff that's been happening near shore? So how do we find out without having to put our dry suits on and dive in this horrible, cold, deep water in the middle of November? Well, the answer, of course, in the modern era is to do it remotely. And so the remote technique for tracking fish is acoustic telemetry. Tags these are little tags that are this is the tag, surgically implanted into the belly of a fish. They're very lightweight compared to the weight of the fish. The fish basically doesn't notice it's in there, and the tag emits a signal which has a code in it that says which fish this is, a coded number, that transmits through sound, which transmits very well through water, and they ping that signal approximately every 2 minutes. In the case of something the size of a lake trout, the tag is big enough to ping for 3 years per tag. So we can follow that fish around on a 2-minute basis for 3 years. Now, we detect them passively. It's not like we can just hear them anytime. We've got to put receivers out in areas where we think lake trout may be. And so when they pass within about a kilometer, about half a mile within the radius of that receiver, the receiver records that signal and puts a timestamp on it. I heard that fish then here. So in 2014, we established an array of receivers, which we call CATOS. This is named after a much, much bigger system that's been established in the Great Lakes, which is GLATOS, the Great Lakes Acoustic Telemetry Observation System. And since, by the way, we started this study, we've also added tagged walleye, tagged sturgeon. This is an array that's available to any species of fish, any study that is acoustically tagging fishes. And we concentrate that early study on spawning sites we knew of. And these were spawning sites, that's Whalen Bay, that's Gordon Landing, and then other areas that looked at transit points between basins or among basins, and then other areas we thought lake trout might be foraging or spending part of their time. So this is how we do it. This is how we track lake trout without having to be out there in the worst of the weather, or even over the winter when everything's iced in. So now with Matthew Fuchsia, a new PhD student, we've started focusing on offshore sites. We started doing some summer work diving and examining these potential spawning sites offshore. We've put receivers next to the spawning sites, and we're basically we're asking the lake trout, where are you in the fall? Are there aggregations of spawning lake trout out here, which might be areas that are the secret to this production of wild lake trout that are surviving? Maybe those little fry that we were seeing along the shorelines in the nearshore areas didn't survive well post-hatch because of any number of issues that we had in the nearshore areas, including, by the way, predators that tend to move nearshore into the spring in order to forage and may have been eating those fish. So there's a number of associated factors between these nearshore and offshore areas that make these offshore areas very interesting. In the interest of time, and I can certainly answer more questions about those, I want to finish up by saying, does any of this research make any difference, right? So, hey, so there's a whole bunch of wild lake trout out there. The role of research, the role of us at the university was to find out, is reproduction occurring? Are there wild lake trout? And then we talk all the time with our colleagues in the state and the federal agencies and just report our results and say, this is what we've got. And then we start this conversation. Well, the conversation from the research standpoint is to write papers. I mean, we talk to our colleagues, but our results get published in scientific journals, and that's the coin of our success, you might say. Well, that's great. Wow, lots of papers about wild lake trout in Lake Champlain and reproduction, etc. But honestly, these are simply read by our colleagues. These are of interest to the scientific community, but they're a little ivory tower, a little obscure, and they're read by researchers elsewhere. What we really need to do is get this back to management on Lake Champlain. So, as I say, we meet regularly. The Lake Champlain Fisheries Technical Committee is comprised of the two state agencies, Fish and Wildlife Service, the USGS, and our colleagues in Quebec. So two countries. And then the policy committee oversees the Lake Champlain Fisheries Committee, technical committee. And then, of course, we've got the anglers, and we've just had a State of the Lake meeting last Saturday, where the anglers are also involved in understanding what's happening with the lake, what is their role in understanding what's happening in the lake, because we do creel surveys to understand their catch, and what should the management agencies be doing with this information about wild fish in this case, and in keeping anglers informed about what's happening. So the first response was, wow, we've got wild fish we really need to know about. The forage fish, as I just explained. And so they supported the idea that maybe the university could start with a new forage fish survey design, and they would ultimately perhaps implement that. So that support was very important, and it was funded by the Lake Champlain Basin Program. They, with us, asked the question, gosh, do we know if these fish are living? And so they, as I mentioned, came together with us with the summer sampling so that we could get information to assess the wild adult lake trout. And then just this year, in fact, only a few months ago, the decision was made to, in fact, drop the number of predators entering the lake by decreasing lake trout stocking, not completely. Let's take a cautionary approach here. Let's drop the lake trout stocking by 30% and see if the wild production can in fact sustain and support that fishable population, but that rising population of wild fish in the lake that will ultimately be, we hope, self-sustaining. And ultimately at some point, we won't need to put all the money and effort into providing what what the lake should be providing naturally, i.e. a self-sustaining population. So good collaboration, management and research working hand in hand to support a self-sustaining ecosystem. So I put questions up there. And what I want to say before I address questions is I really want to acknowledge just that not only the teams out there and the collaboration out there, but the support of the Great Lakes Fishery Commission, who process funding that is provided by our Senator Patrick Leahy is the most incredible advocate of all of the work we do on this lake. And through him, there is an appropriation for Lake Champlain that allows this work to happen, as done through our external collaborators, the Great Lakes Fishery Commission. So, questions? And I will let